Okay, hello everyone. Thank you very much for being here. My name is Downie, and I'm delighted to have the opportunity to talk to you all about some research I've been doing on social association and immune defenses. So, social association has been shown by a lot of folks here and elsewhere to be really important for disease transmission and gut microbiota, right? And the structure of networks. So, we know that connections between individuals and the overall structure of the network is important for how diseases might spread through the network, but it's also important for benign microbes that compose our gut microbiota, such that associations between individuals can be predictive of what their gut microbiota look like, and that individuals who are associated tend to have more similar microbes. And so this has been shown in a variety of different systems, but given that both sort of dangerous microbes that the immune system is guarding against and sort of benign microbes that are actually important for immune system development are affected by networks, it seems reasonable to ask whether we can take one step further and investigate whether social association can predict the similarity of immune defenses in organisms. Now, to study this question, my lab group, the Graham group at Princeton, has been working with rewilded laboratory mice on this question and actually a bunch of other immunological questions. And rewilded lab mice are a great study system because they really have basically most of the hallmarks of a wild organism. They have a natural environment, they have rich microbial exposures, they have a varied diet, they have space for activity and interaction. Basically, the one thing they don't have is predators and competitors, and that's so that we can actually have sample size that we want so we don't lose all our mice to hawks in the middle of the summer. Um, and they do quite well put out into these outdoor enclosures, which you can see here. And because lab mice are a model system, we can do a lot of detailed phenotyping of these mice. So for example, we do extensive immune phenotyping with our collaborators, looking at leukocyte profiles, even T cell memory states. So not just neutrophil abundance, but like more detailed measures, as well as things like cytokine responses against pathogen stimulation. We also do gut microbiome analysis of both bacterial microbiome and fungal microbiome. And so we can really get into the nitty gritty of these molecular phenotypes. But that's not the only thing. So we give each mouse an RFID tag, and we track activity at feeding stations with these RFID tags. So we know whenever a given mouse visits a feeding station. And this actually enables us to build co-association networks, because if we use the rubric that two mice visiting a given station within a given time frame are associated during that time frame, we have a crude measure of co-association that can allow us to build social networks that look something like this. So here are two examples from 2017 in different enclosures. And you can see that these networks are kind of blob-like, so there's not a whole lot of substructure and click formation of our rewilded mice. But there is a lot of variation in association strength. So some mice are more associated with each other. Given that variation, then it seems like we can ask the question whether social association negatively correlates with dissimilarity of immune phenotypes, which is sort of, so we do Bray-Curtis dissimilarity of leukocyte profiles and ask if that negatively correlates with social co-association. And to do this, I did basic Bayesian linear model. And we do indeed find that leukocyte composition dissimilarity does negatively correlate with social co-association in rewilded mice. So dissimilarity is on our y-axis here, and you can see that it negatively correlates with co-association in both of the enclosures that I've done this analysis for. Now you can see it's a very noisy relationship as well, and so there could be a lot of other factors affecting dissimilarity of these phenotypes. But nevertheless, the model is fairly confident there's this negative relationship. So mice that are more socially associated do have more similar immune phenotypes that we can see here. Now, there's a couple possible reasons for this that I'm hoping to explore going forward. So one possibility is that it's really a manifestation of diet, where mice that have a greater association at feeders actually just have more similar diets. Maybe they spend more time at feeders, and so they eat the same thing. Um, and so that's one thing that I'm going to be able to dig into a little bit. Another possibility, of course, is microbes, right? I talked about it at the beginning, like microbes transmitted via networks influence immune defenses. The thing is that we don't see any signs of infections in the mice, and we do check for a wide panel of them. And intriguingly, there's no association between co-association and microbiome similarity, which is not something that's been found in the literature. And that's an intriguing phenomenon that we find here. So maybe it's a third possibility that there's direct behavior immunity links with mice that are more social, inherently having different immune phenotypes matching that behavior profile a priori. There is a particular immune defense that fits with a particular behavior profile, regardless of the influence of diet and microbes. We don't know. That's something that I'm going to be digging into more, and I'm really excited to go into those and share those results later. But for now, I'd like to thank my advisor, Andrea Graham. I'd like to thank my collaborators at NYU and the NIH who did a lot of the immune phenotyping. I'd like to thank my funding sources, and I'd be happy to take any questions that folks may have during the Q&A at the end of the session. Thank you very much.